so thank you especially to Matt for inviting me to talk today and show some of my data. I have started on this only a little over a year ago when I got my K award. So you have seen this slide today already in somewhat of a different iteration, but it's true for, in this case too. So liver disease, obviously we all know that has primary risk factors like obesity, toxins, and alcohol, but it's rare that there's actually only one risk factor actually leading to the disease. There are also host genetics and comorbidities involved. The reason, of course, why we're all here today is the primary risk factor of environmental toxins. Here in Louisville, you've heard about that this morning already too. We are very concerned with environmental toxins since we have Rubber Town in the west area of Louisville. And what's I guess kind of special about this is we have a high density of chemical companies with a lot of employees, but also a lot of residents within 1 mile, and you can see that very nicely in this picture. Also, what we have, and you heard this too, is this unique opportunity of the University of Louisville Biorepositor, which has over 10,000 samples. And this was formed in the 70s when hemangiosarcoma was discovered in a short cohort, small cohort of vinyl chloride workers in Rubber Town. So, and that brings me to vinyl chloride, which is what I'm mostly interested in. The vinyl chloride monomer is produced in about 27 million tons annually, so a huge amount, mostly for PVC production. And it is number 4 on the CDC hazardous substance priority list. Historically associated was vinyl chloride with hemangiosarcoma, but also hepatocellular carcinoma and fibrosis. But what they also found back then, this was actually published in the Tamburo paper from '84, I think it was, where they described that in those biopsies that there was fatty infiltration and/or chronic disease. Back then, they didn't. I don't want to say they didn't pay much attention to it, but this was kind of at a time where NASH, I guess, wasn't really identified as a disease per se. So when Matt published his paper a few years ago, he actually looked at this fatty infiltration in chronic disease and coined the term TASH, toxicant-associated steatohepatitis. He has told you a little bit about this this morning. TASH in the vinyl chloride workers was actually, these vinyl chloride, so these highly exposed vinyl chloride workers had about 84% of steatosis, 80% steatohepatitis, about 55% fibrosis. But the transaminases were normal. So liver damage with vinyl chloride assumes primary injury. So, of course, that would require high occupational exposures. What I'm interested in now is what vinyl chloride does in low doses, because around these production sites or Superfund sites or landfills, there is a large amount of water contamination. And so what we want to know now is, are these low levels, can they be hepatotoxic? And we heard a lot about second hits, and that would be the current setting that we want to explore. So in the beginning, to find kind of a proof of principle type model, we developed an acute two-hit model with chloroethanol, and 24 hours later, we gave a bolus dose of LPS. And what we see here is, I would say, the classic two-hit model. We don't see much liver damage or inflammation in the chloroethanol group, but in combination with LPS, we see a lot of inflammation, necrosis, and this is also reflected in the transaminases and in PCRs of numerous markers for macrophages, monocytes, and neutrophils. What we did see, though, in the chloroethanol group alone was indices of oxidative stress. And again, this was hugely increased in combination with LPS. Then we also did, we stained for tunnel. And interestingly, in the chloroethanol LPS group, though, there was a decrease in tunnel positive cells. And that really meshes nicely with some samples that we have from the Rubbertown residents. This is cytokeratin 18, and I'm sorry if this is pretty small. So what you see here is, I can't even see that myself, this is M30 and this is M65. So this is the M30 is the caspase cleaved version of cytokeratin 18. And this is the M30 to M65 ratio. So this decrease in the M30 to M65 ratio kind of suggests a more necrotic rather than an apoptotic cell death of cells in these Rubbertown patients, which correlates with what we see in the mouse model in the tunnel stain. We also, of course, checked for expression of a lot of pro-inflammatory, pro and anti-inflammatory cytokines, and see increases in a lot of them, mainly in the M1 genes. And then we also looked at lipid accumulation and glycogen depletion, and this is where it gets interesting now. So again, we see huge increases with in the chloroethanol LPS group for oil red O's, which is standing for neutral lipids, but we also see an effect with chloroethanol alone in this group. Also, when we stain for glycogen, we see a decrease with chloroethanol alone, and the livers are completely depleted of glycogen after chloroethanol and LPS. The lipid data are somewhat reflected also in the NEFA and triglyceride measurements. So what explains this effect? And here comes the two-hit model kind of thing back into play. And most of you will know this, but for those that don't, fatty livers respond more to a hit if, fatty livers respond more to a hit. So that means, and we've heard this already a few times, they will be primed. So the inflammatory cells are producing more robust inflammation, and that sensitizes the parenchymal cells, so the hepatocytes, to more cytotoxicity. So to explain those changes in lipid and glycogen metabolism, we looked at carbohydrate and lipid metabolism regulating genes. But really, this is PCR, and it didn't really show us much. It didn't really explain what we were seeing. So in general, with targets in energy metabolism, you have to more look at the flux of the energy through the cell. In order to do so, we went into cell culture, used HepG2 cells, and we used a celomics approach, which you see in figure A. And this is really nice because celomics is a kind of a plate reader with, I think it's 5 automated microscopes. And it gives you what you see in the upper graph here, a result in a well format. And what you see in the lower graph is different, or the single cell response. So what we did here is we stained for mitochondrial membrane potential with TMRM and for cell death with TOTO3. So what you see is this is increasing doses of, increasing concentrations of chloroacetaldehyde. You see that the mitochondrial membrane potential actually goes down before the cells actually die. And this graph shows the same thing. This is 0, this is 80, and this is 200 micromolar of chloroacetaldehyde, where you see here that the mitochondrial membrane potential is actually decreasing, and then at the higher concentrations, the cells are actually dying. Figure B shows a similar result. This is using the seahorse, which can measure oxygen consumption in real time. So what we did here also is increasing concentrations of chloroacetaldehyde, and what we see is a decrease in oxygen consumption. When we uncoupled with FCCP, which should rescue the effect of the rescue the oxygen consumption, which it does in the control, and it does in general, but it still is decreasing, which means that this effect that we're seeing is irreversible. And also, in line with all of this are decreasing ATP levels, which makes sense. The mitochondria are getting hit, so ATP is being depleted. So back to our energy metabolism. So what we know now is that the VC metabolites are actually hitting the mitochondria, therefore decreasing beta oxidation of fatty acids, decreasing aerobic glycolysis, and therefore depleting switching to anaerobic glycolysis, decreasing glycogen levels, and increasing lactate output. But what we also see in what we saw in the NEFA and triglyceride measurements and the OIRED-O is an increase in fat accumulation, which is ATP dependent, which doesn't really make any sense, right, since we have ATP depletion. Just to summarize this again, so we did see liver injury and necrosis, we did see inflammation, and we have direct metabolic effects in this model with mitochondrial dysfunction, switch to anaerobic glycolysis, depletion of ATP, but ATP-dependent lipid storage. So what causes these effects? Here we actually got Ingenuity network analysis. This was highly exposed workers, vinyl chloride exposed workers, and what we saw here was changes in AMPK, AKT, or PepsiK, and we didn't really know at that point if it meant changes up or down. So we decided to look at this in this model. For that, you need to know that mTOR and AMPK are usually activated in opposition, whereas mTOR favors anabolism, means it's activated usually during overfeeding, and AMPK favors catabolism, which means it's usually activated during fasting. So what we see now is in the chloroethanol alone group, we do see an increase in mTOR, but we also see an increase in AMPK. So this is causing somewhat what we call a pseudo-fasted state. So the cells actually think they are fasted, and therefore, the cells are, the cells don't know that they are fasted, so therefore they are storing lipids. So in summary, for this model, chloroethanol is a first hit. We do see sensitization, and as I said, we have liver injury, inflammation, and direct metabolic effects causing a pseudo-fasted state of the cells. The problem with this model is these are skinny mice. And the reason why this is a problem is, well, not necessarily a problem, but what we want to actually know is, with the obesity epidemic going on in the US, we want to know if steatohepatitis or fatty liver due to overfeeding, overeating of the general population will actually cause vinyl chloride to be a second hit. And so we have above 30% of obesity in the States, but this doesn't mean it's 30% of obesity in all areas of the population, but it's also connected to the socioeconomic state. So of course, in areas around, like production site like that, around Rubber Town, we will have almost 100% of people being at least overweight, if not obese. So for that, we created another two-hit model. In this case, we did high-fat diet and chloroethanol. The high-fat diet was fed to the animals for 10 weeks. And some of these data are really fresh off the press. And so what we see here is a robust increase in transaminases, a robust increase in inflammation, and even in fat. We see, I think, also, again, more of a robust increase in the chloroethanol and high-fat diet fed mice and the pro-inflammatory cytokines. And we are still working on what's going on with the energy metabolism. And obviously, the VC metabolites are doing the same here. So again, the mitochondria are being hit. So we looked at the genes of carbohydrate and lipid metabolism again. And in this case, we actually did see some promising differences. But we have not looked into what's going on with the mTOR AMPK paradoxon, I guess. So in summary, for the energy metabolism, chloroethanol can also be a second hit as compared to our first model being a first hit, causing, again, liver injury, inflammation, direct metabolic effects. But we were also interested in ER stress. We looked at that also in the acute model with the chloroethanol and LPS, but we didn't see any effects here. But in the chronic study with the high-fat diet, we did see robust increases in genes that are attached to, well, are in contact with ER stress. So we also tried to model that in vitro. And again, we used HepG2 cells. And we were actually really happy to see this. So here what we did is expose the cells to palmitic acid and then expose them to chloroacetaldehyde. And we see pretty much the same effects that we see in vivo. This is a classic lipotoxicity assay. What we thought was really cool is we used a very low dose of 20 micromolar. If I can bring back this slide, and you remember, at 20 micromolar, we didn't really see any effects on oxygen consumption, cell death, mitochondrial stress, or ATP depletion at all. So these data are actually really interesting. So as a summary for the high-fat diet and chloroethanol, again, as I said, chloroethanol is a second hit, causes injury and necrosis, inflammation, metabolic effects, and it does also cause ER stress. Then something else that we are also working on right now, but I'm not going to show you data really today, is the importance of OXLAMs. This has also been shown in serum metabolomic analysis in vinyl chloride workers. Craig McLean mentioned it this morning that OXLAM worsen alcoholic liver disease. And a colleague of ours, Irina Kirpich, showed that OXLAMs actually decrease mitochondrial respiration. So for us, this will be really interesting because this could actually explain some of the metabolic effects we see. So we actually did a study where we used a diet high in linoleic acid. But as I said, I'm not going to show you, in the interest of time, I'm not going to show you any data of it. But this will be really interesting to see the metabolic effects. So as an overall summary, vinyl chloride and its metabolites do sensitize the liver and or it damage the sensitized liver, as you wish. It works in both directions. And with that, I want to thank my lab, Gavin Artiel's lab, my collaborators, Craig McLean and Matt Cave, and everybody that was involved. Thank you.
We have time for questions. Go ahead.
I just have a very simple question, which was, in your mouse study, I just couldn't quickly gather, what was the dose that you used?
Of the chloroethanol?
Yeah.
50, 50 micrograms per, I don't know, milligrams per kilogram. We based that on a study by Cafalia et al. From '84, where they used that dose for, I think, 5 days to see effects.
I have a comment and a question. The comment is, this is really cool. The question is, how much do you, in the high-fat diet scenario, I'm trying to think back to Dr. McLean's talk this morning, how much do you think that maybe portal endotoxemia or whatnot will be a critical component for kind of sensitizing the mice or whatnot to VC exposure? Or is it more about, if you could make the liver fatty, make the hepatocytes fatty in the absence of endotoxemia, would you get the same sensitization?
That's a good question. And I don't know. I could imagine that it's that plays a major role. Yeah, that would be one thing to test. The thing about vinyl chloride and its metabolites, there is really nothing out there, which is kind of cool in a way, but also not, because I have to kind of do everything ourselves. Yes.
So quick question, with your pseudo fasting that you see, with treatment, you have a significant depletion of glycogen, right, in the liver.
Mm-hmm.
Do you see any changes in liver to body weight ratio based on the glycogen depletion data that you have?
We did not see significant differences, no.
So liver weight doesn't change significantly?
In the high-fat diet, it goes up,
Yeah.
but in the acute study, we don't see any differences. I mean, nothing significant, no.
So do you estimate more or less what percentage of glycogen depletion you get in comparison to, say, a normal fed animal? Do you have a sense of what?
No, I don't
that
know.
might be?
I don't know that. I'm sorry.